This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology. This is a special episode recorded on April 21st, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Today we're recording at 50 years of reverse transcriptase. This is a meeting being held at Cold Spring Harbor Laboratory. So two groups independently discovered that enzyme in 1970, 52 years ago. And we've gathered three individuals to chat about that tonight at this meeting. And we're lucky to have one of the discoverers here from Pasadena, California, David Baltimore. Welcome.
Hello.
David was previously on TWiV 100. Which was a long time ago. In fact, everybody here tonight has been on an episode of TWiV. Next to David from Boston, Massachusetts, John Coffin. Welcome back.
Hello, everybody.
This is John's third TWiV. How about that? And at the end of the table from New York, Harold Varmus. Welcome back.
Thank you, Vince.
Harold was on number 400. And this is going to be 890-something probably, so we're approaching episode 1,000. So I wanted to start with you, David, and transport yourself back to the 60s and 70s and give us a sense of how the work you were doing at the time led to the experiments to reveal reverse transcriptase.
Well, if I go back to that time, what I was concerned about was how RNA viruses run their business. What is the genetic system within which RNA viruses live? And I had been working on that kind of issue ever since I started as a graduate student in 1960, and this was now 1970. And we had put in place much of what we wanted to know about how RNA viruses work, but there was one group of viruses that didn't fit, and that was the RNA tumor viruses, a group of RNA viruses that cause cancer. And Howard Temin had, 10 years before that, suggested that they had to put their information in something more permanent than RNA, because RNA degraded very easily. And that had to be DNA. But that violated the central dogma, which said that information flows in biology from DNA to RNA to protein, not back. Now, actually, the step back from RNA to DNA was not really very complicated. DNA and RNA are chemically very similar, and moving information from one to the other in either direction is energetically very straightforward and is mechanistically very straightforward. If you can copy DNA into RNA, you can copy RNA into DNA. But it had never been seen. And so this notion had solidified into our thinking. So how do these viruses work, was the question. And what I realized was that if I did what I love to do, which was to bring biochemistry to the problem, that maybe a solution could be found. And that would be if the enzyme that carried out this transfer of information, if it happened, if that enzyme was in the virus particle. And there was reason to think that it might be. There were a number of other viruses that carried this kind of transformational molecule in the virus particle. Viruses that used one strand or the other for their information and transferred it back and forth. Viruses that had double-stranded RNA as their genome, viruses that had DNA as their genome. All of those were precedents for what we were thinking about. And so I set about to look for that enzyme.
So, John, you were a student, a PhD student with Howard Temin, who unfortunately isn't here. But maybe you can tell us what was his thinking leading up to
Yes, when I was a student, he had already, I started in the lab in 1967. Howard had come up with this idea, as David said, based on a sort of an indirect genetic evidence. And he observed that cells transformed by a couple of different strains of virus into cancer cells had different shapes. They looked different in culture. And that was a very heritable property due to the virus, not the cell. And from that, he basically intuited that the virus must be making a permanent genetic change. And he then went on, and I believe, although he didn't say this, he's more cautious than this in this paper, but I believe that he really had pretty much the whole construct in mind at that point, that the virus RNA, the genome of retroviruses, or sarcoma virus, this particular one, had been well established to be RNA at that point. Not that long before, actually, but had been well established to be RNA. But that RNA must be converted to DNA, and the DNA must somehow then become permanently associated, integrated, is what we would think, with the cell DNA. And as David said, this is something that people were very much unwilling to believe. And Howard published several papers of various kinds purporting to show that this was the case, trying to get proof of that. He showed, for example, that virus production from infected cells was sensitive to actinomycin D, a drug which more or less specifically inhibits DNA-directed RNA synthesis, into which most RNA viruses, although not all, were known to be completely resistant. And he published a hybridization experiment that showed very, very small numbers of counts per minute in very large amounts of DNA, and with quite a high background. And so people who didn't want to believe this certainly had license not to believe based on these studies. And he presented experiments like this, with some frequency at meetings like Cold Spring Harbor, very frequently. I remember very well he would come back from those meetings actually quite depressed, because really nobody believed him. People were skeptical, almost to the point of laughing at him, but probably not quite that bad. But there was extreme skepticism being voiced at meetings. In some ways, I've always thought if you're convinced that you're right and you want to make a big splash, you do a lot of experiments that sort of give the right result, but aren't really going to convince everybody. You do that for 10 years, and by the time you actually can do the dramatic killer experiment to show you're right, everybody will say, wow, and you'll be lionized, and that's actually what happened, but it's a tough strategy to carry out over 10 years.
You also have to recognize that Howard Temin was one of the most admirable, smartest, most thoughtful people I ever knew.
Absolutely.
So it's not as if he was some crank.
Right.
But this kind of story has repeated itself in science. I'm thinking of Stan Prusner, right? No one believed him for 10 years about prions. Right? So, Harold, what were you doing in 1970?
Well, I bring a rather different perspective to this, even though the three of us are more or less the same age. I was much less far along in my career as a scientist, and David was an assistant professor. John was at least coming close to the end of his graduate career, working in the lab of a leading virologist. I was someone who had gone to medical school after abandoning a graduate career in literature. I was obligated by the U.S. government to do something to help the country during the Vietnam War, and the one thing that was available to me other than jail or Canada was the NIH. And I was fortunate to get accepted there because I hadn't done laboratory work of any consequence before. But I ended up loving doing quite fundamental science, studying how genes in bacteria are regulated, and yet had come to feel that that as a trained physician interested in medicine as a long-term career, if I were going to follow up on my newfound interest in doing laboratory work, I should do so in some context that feels medical. And I had been taking courses at the NIH where I was working, and I had learned that people who were trying to study cancer had some exciting things to show if they focused on viruses. You have to remember, those of you listening to this, that at that time it was very difficult to study genes that come from human cells. There was a lot of reason to believe that cancer was in some sense a genetic disease. It might involve aberrations of genes that were inherited in some kind of abnormal form or became abnormal during life in individual cells. But there was no easy way to try to figure out what those changes might be and what genes were involved. But there were viruses, some carrying DNA that we haven't talked about, some carrying RNA, as we've begun to discuss. I had read in the courses I was taking on the NIH campus about Howard Temin's striking ideas, for which there was, I have to say, incomplete evidence. I had learned one pretty powerful trick as a young scientist trying to learn something about working in a laboratory, and that is that you can measure nucleic acid molecules individually by using tools that rely on base pairing between molecules to say, is the companion, is a molecule that's similar to the one I'm working with actually found in the solution of the cells I'm trying to study, technique called molecular hybridization. And I became convinced that I could try to determine whether Howard Temin was right by looking for the molecules, the DNA that was related to the viral genome of these RNA tumor viruses. So when the great experiments were done in the Temin and Baltimore labs, I was just getting set to head to the West Coast to study this problem with a young virologist who had similar experience to my own, not yet well known. And so the setting I was in was one of surprise that the problem I wanted to work on may, in the minds of many, have been solved by these experiments.
Okay. So, David, can you tell us what was the experiment that you did?
The experiment that I did was to find a stock of virus particles and to treat them as I would an enzyme, a protein that carries out a biochemical reaction. The biochemical reaction in this case was the synthesis of DNA. And so I assayed them for the synthesis of DNA. And I was able to show, after literally a couple of days' work, that virus particles of the class of RNA tumor viruses would synthesize DNA, and that synthesis of DNA had all the properties of a copying of RNA into DNA. And I put that together in a paper, and it was sufficient, particularly along with the similar work from Howard Temin's lab, to convince the scientific community that this hypothesis was extremely likely to explain the nature of these viruses.
Was it difficult to get the paper published?
It was not difficult to get the paper published. I sent it to Nature. They sent back right away and said, we'd be happy to publish it. I said, hold on, Howard Temin has similar data, because I had talked to Howard about it in the interim, and they held it up for a week, and then they published it in the following week. They said they reviewed it. I've never met anybody who said it was he or she was the reviewer.
It's a good thing. If you did it today, it would take you a year probably.
Who
to get
knows?
it published. And is it correct that you also presented it that summer here at Cold Spring Harbor?
That's right. So this was May 1970 when we did the experiments, and the paper was sent in early June. Traditionally in June, the big symposium that sort of defines this year's data in molecular biology was being held at Cold Spring Harbor. And so they, having heard about the work, they said, why don't you come and present it, even though it really didn't fit the particular topic of that year.
I'm curious, anybody at that meeting here in the audience? Hung. Wow, 2 people. Anyone else? 2 people. That's not bad.
Harold,
I was there on my way to California. I stopped
Really?
off here.
Oh.
And then to a Gordon conference in New Hampshire where David also spoke.
yeah.
So is that the first you'd heard of it, or had you heard of it?
No, I had read about it because there had been some notice of the talk that Howard Temin had given at the MD Anderson Symposium a couple of weeks earlier.
Okay. So what was your reaction to it when you heard this?
My reaction was, well, what am I going to do? Is everybody going to believe this? And, I was very happy about heading for California. I had my new wife with me, I had my fishing rod. I knew I was going to have a good time in California, but I didn't know what I was going to do. But we drive across the country and take a lot of stops. You have time to think. And as I thought, and I reflected on the talks that I heard having been given especially at the Gordon conference on animal cells and viruses, I realized that, hey, there was still a hell of a lot to do. These guys had found the enzyme. That made everybody much more optimistic about the idea being right. But what you want to know is, can you actually find the DNA version of the viral genome? What does it look like? Where is it made? How is it made? Does it get integrated into the host chromosomes the way people thought it might? How does it get expressed? And, more questions appeared the more I thought about it. And I realized that the virus was not, the enzyme was a great discovery, and it answered a lot of questions, but it opened a lot of questions, and equally importantly, provided the technology, the tool that would allow us to try to answer the questions with what this one-trick pony had to offer. That is, I would be able to use it to make radioactive DNA that I could use as a reagent in hybridization experiments that would allow me to look for these things and try to characterize what the enzyme was actually doing when the real thing happened, not when David put the virus in a solution and tried to run an enzyme reaction, but when the virus actually entered a cell and caused infection and made cells behave in a new way. Because that, I found replication of viruses incredibly interesting, but one of the big motivating facts for me was, how does this virus cause cancer?
Yeah. So, John, were you in the Coffin lab at the time?
The Temin lab, yes.
Temin lab, sorry.
There was no Coffin lab yet at the time.
Temin lab.
There is now. There wasn't.
You were in the lab, and
I was in the lab.
and these experiments were being done at the same time.
Experiments were being done. In fact, as I presented yesterday, I played a very important role in those experiments. The virus required a non-ionic detergent to break it open, so that the nucleotides could get in, and the detergent of choice was NP40, Nanodet P40, and I had the only bottle of that in the lab. So I loaned it to Satoshi Mizutani, who was the postdoc who Howard had hired it. I think because of his training in nucleic acid chemistry, and so that he could do things. The lab didn't know any biochemistry, so that was foreign to us, especially nucleic acid chemistry.
You said that the other day, and I'd never heard before the idea that Mizutani was hired by Howard because of those strengths, and because Howard wanted to apply biochemistry. But that's your guess.
That's my guess. I don't have any direct evidence for that, but it seemed to make a lot of sense that I think he was inspired by learning about Alice's your
The work that my wife and I had done
on vesicular stomatitis virus, showing that virus contained an enzyme capable of copying RNA into RNA. But that sort of set the stage for, because the principle was that if this was going to be true, if it was going to be true that RNA was being copied into DNA, there had to be an enzyme to do it. And nobody had ever identified an enzyme in cells that could do it, which doesn't mean it wasn't there. Mm-hmm. And it would have been very hard to do that. But the idea that it could be in the virus makes everything all of a sudden extremely simple. So this was an experiment that David did, Howard did, and particularly after David's presentation, and certainly after the papers came out, probably 100 other labs in the country did, and they did it in a much easier way than David, because almost all of them already had a good stock of virus ready to go.
It's actually surprising how quickly many other labs did do the experiment. Some rushed out of the meeting and went to their lab that night. Others had been thinking about looking for the enzyme. And by the time I got to New Hampshire a couple of weeks after the Cold Spring Harbor meeting, there must have been 6 speakers, including my own lab mate, Mike Bishop, and quite a few others who had done quite a few experiments and begun to characterize the protein, the DNA being made by the enzyme.
Well, this is a relatively straightforward experiment. And you had been doing it for some time with VSV, correct?
It's a canonical experiment. I had been doing exactly that sort of experiment for 10 years.
And one of the staggering things we saw last night was David's lab notebook page. And it has a few ingredients lined up with some scratch marks indicating how much of each material he was putting in. But it was a pretty simple experiment compared to some of the things we do these days. But it had about 1,000 times more meaning than most of the things we do with much more elaborate reagents today.
I heard you say on another podcast, it just took you 2 weeks to do the experiments. But that's true, because we knew where to look.
Yeah.
Right? That's a great way of putting it.
Yeah.
So you were aware that this was happening in Howard's lab at the same time, or no?
No, I was not aware of it. I had done experiments in my lab. I did them all myself. And then I called Howard because I had known Howard from earlier interactions. And I wanted him to know that we had basically provided the evidence for what he'd been saying for so many years. And that's when I found out that he was doing similar experiments.
But you also made an attempt to publish it at the same time, right?
They did. They were published.
Yeah, they were. Time. Yeah, I did.
But that was an active act. You didn't. It wasn't a random thing that just happened, right?
No, actually, I had either already sent off or was about to send off the paper to Nature, and I asked Nature to hold it.
I think that's important as a kind of lesson to contemporary science, that you have to respect each other. And I think in the end, it strengthened the case that 2 independent labs did it, right?
No question.
Absolutely.
When 2 labs have both done an experiment coming from different points of view and different strengths, it has much more solidity to it. And in this case, there was never any question that the experiments worked the way we said they did, and that the implications were very clear. There was a lot more work to be done, as Harold has said, but with 2 different labs doing the same thing, there was very little doubt.
So was that the last experiment you ever did?
No. No, I actually continued to try to do experiments until 1970. That was 1970, until 1975, or 1976 maybe. And I remember the last experiment I tried to do. It was a recombinant DNA experiment. It was fairly complicated, required growing up bacteria. And all I did was to contaminate the bacteria I was growing up. And one of the postdocs then in my lab said, just go back to your office and let me finish this. And that was the last time.
I think I heard that story. Yeah.
Yes.
Because when I arrived in your lab in '79, you were not in the lab
I was
anymore.
not in the lab.
Yeah. Where were the RT experiments done?
In MIT.
MIT?
Yeah.
Okay. Harold, I really like this idea that at first you think, oh, it's done, I have nothing to do, but then you just think about it and there's obviously more. It's very important, right? Because if you think, and you had a whole time going across the country to think, you could...
Well, not all these ideas came to me during that drive, but I think one of the things that's illustrated by this meeting is what the utility and significance of the discovery of this enzyme were, because we now, thanks to the existence of the enzyme to make certain kinds of probes, to look for other things, thanks to the precedent of establishing that reverse transcription actually does occur in nature. So many things have emerged from that. Many things we use routinely in biotechnology. New ideas about how other viruses might grow, how so-called movable elements might move in cells. We now know that a large fraction of our genomes are synthesized by reverse transcription sometime in the past. So it's influenced in a dramatic way the way we perceive living systems to work.
John, where did the name come from?
Reverse transcriptase? Well, it's in the trivia quiz that we put out.
Oh.
I do know the answer to that, but I'm going to save that until the end of the meeting.
So there's a trivia quiz in the program. Yeah, 10 questions.
Trivia,
Who named
is a
reverse
tradition of the retrovirus meeting.
So that means you can't answer it? Okay.
I can answer it, but I won't answer it right now. You'll have to wait until Saturday, and all will be revealed.
Okay.
You've learned stubbornness from Howard Temmer.
Yes, that too. Yes.
So David, when you and Howard discovered this enzyme, did you really think it was unique to retroviruses?
We had no idea. There was certainly no other virus class that had the properties of the RNA tumor viruses. So there was no obvious candidate. And actually, the candidate which came by later was 10 years later, was HIV, was the virus that causes AIDS. And that turned out to be of the same class of viruses. Mm-hmm. But there weren't many others. There were a few. There were a group of viruses called slow viruses, just because they take a long time to do their work.
When did hepatitis B virus reveal its RT?
Later.
80s, maybe?
Yeah, I don't know when that was.
But that,
80s.
But
that is an important point. One of the things we've explored at this meeting is the fact that there are other viruses that don't look like they use reverse transcription because they have DNA in their virus particles.
That's
right. Yet the virus life cycle does include copying the RNA that's made from that DNA to serve as a template for making DNA in the virus. So from an evolutionary standpoint, it brings you into very deep water where you begin to think about what nucleic acid was actually the first nucleic acid and how much use nature made of RNA templates to make new DNAs. It's a powerful way to operate. It gives cells more flexibility.
Of course, now we know that RT is in bacteria and archaea and eukaryotes.
Mm-hmm. Yeah.
It probably precedes viruses, right?
It may well.
But you had no sense of that at the time.
Where will we get that from? No.
It's easy to look back and say.
The other thing is that it was already so dramatic that we had found this that we didn't need to add to the drama.
Well, part of the reason I dwell on it is because I think that the thinking was this was a unique enzyme, and that's why you looked for it in virus particles. But what if you knew it was in cells? Maybe you wouldn't have looked for it in virus particles. So
yeah. I mean, if it were a commonly known enzyme, we would have just postulated its involvement. It wouldn't have been a big deal.
Yeah.
Indeed, the question you're raising, Vince, is pertinent. It's possible that even with the discovery as described, that The enzyme they discovered was from cells originally, and just was packaged up in these viruses for some reason, and maybe even contorted in some way by the presence of viral proteins or viral RNA to behave as an RNA templated rather than a DNA templated polymerase.
I think those are wonderful things to think about. In fact, many people think reverse transcriptase is one of the most ancient enzymes and allowed the creation of the DNA world from the RNA world, right?
Well,
I think that's what people think. I think applying Occam's razor to events that have taken place over billions of years, I think is a bit misleading. The enzyme, it could have gone back and forth through multiple evolutionary steps and become a DNA polymerase that was originally maybe a reverse transcriptase and making the original steps from the RNA world to the DNA world, but then evolved into a DNA polymerase and then re-evolved back into and there's nothing that I think says that can't be the case.
But you're right. It's a nice story if
you're right. It's a nice story.
RNA to DNA. Yeah,
it's nice to have these simple straight arrows covering billions of years of evolution.
Well, I talk to Eugene Koonin a lot, and he likes to do that.
He does.
So, David, after this discovery, what was the impact on the direction of your research?
The major impact was that I said to myself, this was the first experiment I had ever done with a cancer-causing virus that had anything to do with cancer. I sort of put my mark down in this field. It would be very exciting to see if we could learn more about the cancer problem. And so I went in search of a cancer-inducing system that had the properties that would enable me to do the kind of work I wanted to do. And that first of all meant working with mice. And second of all meant working with something that grew pretty quickly because I didn't want to spend the rest of my life just growing virus stocks. Something which caused cancer in a very well-defined way so that I felt I had a handle on the biology. And I discovered that. The Abelson mouse leukemia virus, a virus which was very little known to the scientific community, but had been found by this guy, Herbert Abelson, at NIH. And it had all the properties I wanted. It caused cancer quickly. It caused it in cells and culture. It could be quantitated. And we then spent the next 10 years working on it.
And if I'm not mistaken, that got you into the immune system.
That got me into the immune system. Because it turned out that virus was actually turning cells of the immune system into cancer cells. It was an entree into immunology, which was what I spent the rest of my career doing.
And when I arrived, by the way, there were people in your lab working on understanding the mechanics of reverse transcription. I remember strong stop DNA, hearing that all the time. Yeah, and so forth. What about you, John? Did it do anything to your direction?
Oh yeah, no. I spent a lot of my career, particularly my early career as an independent researcher at Tufts University, on experiments to understand some of the mechanistic details of the process of DNA synthesis. We discovered, for example, that based on some preliminary data that had come from some of the people in this room, actually, we came up with a model for how reverse transcriptase might be able to make a full-length molecule and get around some problems that are involved with DNA synthesis, and that was by synthesizing the DNA to one end of the genome and then using a terminally redundant sequence to transfer the growing DNA chain to the other end and allowing completion. I never ever was able to figure out what the remaining steps were and how you actually made the other strand, and that actually that was actually left to David's lab to work that out, and Harold as well. But then I did some other work, for example, on how how recombination works, and on how viruses transduce oncogenes, and so those are all consequences that follow very, very directly from how reverse transcriptase works.
And Harold, you said you arrived in San Francisco.
There's another fold to the story.
Yeah.
That is that unlike these guys, I had a medical degree, and I came to California hoping to learn more about my interest in and ability in science for another couple of years by pursuing two big questions: how do these interesting viruses grow, and how do they cause cancer? But I also knew that it might not work out. I saw at the meetings I attended on the way west that a lot of people were suddenly very interested in these viruses, and it was going to be kind of competitive. And one of the things I did a few months after arriving in San Francisco was to go to the chief of the Department of Medicine and say, could I fit in here if the science doesn't work out? And he sent me off to become a specialist, and that didn't work out very well either. And after several fairly unsuccessful months in trying to characterize the DNA being synthesized in newly infected cells, we formed a collaboration with a scientist who had been in this field for a long time, Peter Vode and his trainees. Peter's happily here today. We were introduced to some biological properties of both viruses and the host cells that allowed us to do two things that proved to be important. One is to work with ideal host cells for studying the steps by which viral DNA is being made by reverse transcription when a virus enters a cell, and to characterize its properties, its physical structure, the organization of its genes, the repeat elements that are too complicated to talk about on the radio without slides, but are integral to the way in which the virus grows. And I was also introduced, as was my partner in this science, Michael Bishop, who is speaking at this meeting tomorrow, to mutants of the virus we were working with, the chicken virus called Rouse sarcoma virus, that might allow us to explore the nature of the gene that the viral geneticist has been working on and had been able to assign a transformation, a cell-altering function to. And we saw ways to begin to look at this gene, the kind of protein it makes, where it came from, importantly, and why the virus was carrying it. And so these two, I suddenly saw within a year or two, not immediately when reverse transcriptase was announced, but as we marched along in some reasonable path, I saw there were ways to explore how the virus grew and how it caused cancer in ways that would be different from what everyone else seemed to be doing, and might provide both pleasure and rewards.
I'm curious, in the audience, who has not used reverse transcriptase in their research? Yeah, not a single hand. See, I learned not to ask the other question last time. I asked who works on retroviruses. That's the wrong thing to ask at a meeting like this, right? So, David, let's explore the impact of RT on viruses in general, our understanding of virus biology.
Well, it sort of closed a system that you could use to pigeonhole basically all viruses that are RNA viruses that had DNA as their genetic information, viruses that carried enzymes in the virus particle, viruses that didn't, and you could classify them into 7 different styles of growth. And that pretty well encompassed the whole kingdom of viruses. And viruses really are, in a sense, a kingdom. They're quite separate from the rest of the organisms that we deal with because they're parasites, because they can only grow inside cells. And I wrote a little article about that which has become the way that people classify viruses in their simplicity, in just these few different styles of living. And there are a couple of viruses that don't fit, and there are hybrids, and there's all sorts of footnotes. But that was very satisfying, that you could bring together all of virology into a pretty simple set of patterns.
I use that in my course every year.
So do I. I think the Baltimore classification is still the standard for teaching viruses, because it tells you that with a very small amount of limited information, you can make a very compelling prediction about what the virus replication cycle looks like. And if you're interested in studying that virus, and it's a brand new virus, that really points you exactly where to look for further study.
But also illustrates the power of classification by function rather than classification by shape.
Right.
That's right.
Yeah.
That's right.
Yep.
So I call it the Baltimore scheme, which many people do. And I have a picture of it, and I have a picture of you catching a big fish next to it. so they understand who you are. And once I heard a student say to another, "You have to learn this. Racaniello adores this Baltimore scheme." But I think that's a really important point. Of course, RT has been a tool for every other virus as well, right? And every time I hear a SARS-CoV-2 RT-PCR test, I think, wow, where would we be? Right, without it. John, what about broader impact on biology?
Well, I mean, reverse transcription has been discovered to be important in many biological processes. I mean, the most compelling, perhaps, in normal eukaryotic cells is in telomerase, that the enzyme that prevents chromosomes from getting shorter and shorter and shorter with every replication cycle, at least in some cells. In other cells, the telomerase is inactive, and they do get shorter with every replication cycle. And also in various kinds of sort of, in a way, what certainly at the time we would have considered to be very oddball virology, or very oddball biology, rather. Mobile introns that move around cells, and in a number of other viruses where it wouldn't be obvious actually from looking at the virus, like hepatitis B virus, cauliflower, the plant virus, cauliflower mosaic virus, and a few others that also use exactly the same reverse transcription. They use reverse transcription sometimes in rather different ways, and also the fact that our genome is about 40 or so percent consisting of elements that were originally put there by reverse transcription. Most of those are actually a very different kind of element, not a virus. Some of them are viruses. About 20% of those are actually endogenous proviruses. In fact, they're still something like 80. That makes something like 80,000 different bits of DNA, and so I like to tell the students there are more viruses in us. There are only about 20,000 genes, so there are more viruses in us than there is us in us. And so really reverse transcriptases form more of our genome in a way than almost anything else by and large.
So what does not have reverse transcriptase in its genome? Do we know?
Well, bacteria use it very, very differently. I don't know if all bacteria actually have been identified as having some reverse transcriptase. I don't think all have. Some certainly have.
But all multicellular organisms?
Well, also in yeast.
Yeah.
Yeast
as well, yeah.
Harold, what do you think about broader impacts?
Well, we don't want to underestimate the effect this has had on research. I mean, your rhetorical question to the audience was indicative of the fact that virtually everything we do in science today is dependent on reverse transcription. And whether it's copying messenger RNA to make cDNAs or making the first step in amplification process, the Human Genome Project depended heavily on it. But I just think things would have moved much more slowly over the last 50 years if we didn't have reverse transcriptase as a tool to help us study all kinds of things.
PCR assays for HIV and for coronavirus don't work without it.
That's right.
I'll tell you a story. that illustrates how you should not do science. So years ago, the first trip I made went to China. And they had invited me to Wuhan. And in Wuhan, there were a lot of virologists. And one of them, a young virologist, came to me. He said, "I need some advice." I said, "Well, I'm happy to hear what you're doing." He said, "I've been asked to sequence an RNA virus, but I don't know how to sequence RNA. Can you help me understand how to sequence RNA?" Well, there was no good way to sequence RNA at that time, certainly that I knew about. So I said to him, well, the way to do it is to turn it into DNA, copy it into DNA, and then sequence the DNA, because we know how to sequence DNA. He said, "I can't do that." I said, "Why can't you do that?" He said, "Because I've been told that they sequence DNA in Beijing. In Wuhan, we sequence RNA."
Right. It's not a way to
to do science.
So politics shouldn't be messing in science. Is that what you're saying?
That's what I'm saying.
I think Wuhan no longer has that rule.
No, I think that they've outgrown that. Right.
They have other problems.
Yeah. So I wanted to return to the name, even though you're not going to tell me who coined it. So you certainly did not coin the name reverse transcriptase.
No, since I don't know the answer that John is going to give,
go ahead and guess.
what I know is that it was originated in Nature magazine.
Okay.
And in an article, the News and Views, I guess, that they wrote, they coined the word reverse transcriptase, but he may know more precisely who it was.
I happened to be sitting next to this person at a meeting.
Well, I wasn't. So.
And asked this person point blank and got a confession.
Well, you do have special knowledge.
But you're okay with the name, right? It's a good name?
Oh, I think it's wonderful. I mean, because calling it RNA-dependent DNA polymerase is a mouthful. And reverse transcriptase is much more
Okay.
indicative.
So neither you...
Distinctive.
It is very good. No,
Right.
RNA-dependent DNA polymerase, DNA-dependent DNA polymerase.
Right.
Who can keep track?
Right.
But, you mentioned reversal of the dogma. Isn't it correct that Francis Crick said, "I never said that"?
Yeah.
That's right.
Yeah, that's correct.
Francis, the dogma is written where dogmas are written as DNA transfers information to RNA, transfer information to protein. But Francis said, I never took seriously that it couldn't go back from RNA to DNA. And if you know the chemistry, I'm on Francis's side. It's pretty straightforward chemistry. We're not violating any principles of energetics or physics or anything else.
So I think it was created as a dogma, not only because Francis said it, but because that's the way everybody took it. That's what almost everybody took it to mean.
I do think it's a useful teaching point. That is, to ask a student, what should the central dogma be, and why would it be the case that protein would be hard to extract information from and to create a genome.
But by calling it reverse transcriptase, you're implying that there is a normal flow of information.
Right, and there's no question there is a normal flow.
There was another reason for doing that too, though, and that is in RNA viruses, like the ones David had been working on, the enzyme that replicates them is referred to as transcriptase. The enzyme that replicates them or copies them, depending on the
And there's a historical
genome.
precedent of knowing one direction first and the other. And if you weighed the amount of RNA made from DNA as opposed to DNA made from RNA, you'd find there's a discrepancy.
Neither you nor Howard patented the enzyme.
Right.
Why is that?
Well, I can't speak for Howard, but I can speak for me. It never occurred to me. Patenting was not on my mind. And I guess it was not on anybody's mind at MIT at that time, because nobody came to me and said, we think you should patent this, or do you want to think about patenting it? And I must say, I'm perfectly glad that we never did patent it, because that's the reason everybody's using it, because it's free and freely available to anybody who wants it.
But it's a question that we ought to think about, because I don't think it reaches the rules of patentability. It's a discovery, not an invention.
Okay.
Well, you have to predict a use for it.
Yeah,
And also a process for obtaining it, and so on. I think you could write a patent could be written around that, although probably wouldn't be a very strong one.
But you could. I mean, I think it'd be easy to evade the use patents and the methods patent.
Lots of discoveries are patented in that way.
A thoughtful patent lawyer could have found a way of patenting the essence of the discovery, but we never.
Well, if somebody had thought of using it to make cDNA probes to look for, to test for the presence of viruses and things like that, it would have been very straightforward.
9 years later, when I came to your lab and used reverse transcriptase to make a DNA copy of polio, we patented that.
Well, you can. I mean, and the Supreme Court's decision on the patentability of DNA admits that.
Yeah.
If that is an invention, you make something which is not present in nature, namely a cDNA. Now, it's very easy to change the nature of the cDNA that you make and avoid the patent restrictions, but
whatever happened to that patent?
Well, it expired. It's over 17 years. I don't think
did you make things?
I don't think we made anything. But it cost a lot of money for MIT to.
Probably $30,000.
Yeah, I don't think we made anything.
I'm pretty sure that, like David Howard never have occurred to him at the time to do that.
Yeah, well,
it certainly wasn't. He certainly wasn't opposed to patents. He patented some things later on himself. But
I'm reminded of Jonas Salk, right? They asked him, "Why didn't you patent the polio vaccine?" And you know what he said, right?
Yes, we do.
Could you patent the sun? Which is not really the point, but it's a good sentiment, I think. And you're right; it probably doesn't restrict the distribution of it. I wanted to ask one last question of you, David. I have this image of you thinking of an experiment and doing it, taking risks. I just don't see today that we can do that kind of work, and I think we should be able to. And funding is very difficult. So, is there anything we can do about that?
Well, it's hard to imagine a question that could be answered with as simple an experiment as we did then, coming up today. We've sort of taken a lot of maybe all of the low-hanging fruit from our science, and it's pretty complicated. We've, for instance, spent the last few years trying to understand what small RNAs called microRNAs are doing in cells. It takes very complicated experiments to test that kind of question. But let me change the question around. Should people have the freedom to do that? And the answer is, as far as I'm concerned, absolutely yes. And it means I was running my own laboratory. I had my own budget. I got the money to do other things because nobody had ever thought of doing this, but I could do that. And I was actually I was an associate professor at MIT at the time. I think it's important that young people get the independence that allows them to try things that may look stupid, that may look unlikely, that may be flyers. And people say, well, do you want to risk doing an experiment that doesn't work? I'm not sure I believe there is such an experiment that doesn't work, because learning that something isn't true is often as important as learning something that is true. I do this little Wordle puzzle from the New York Times every day. And the first thing you do is you learn all the things that aren't right. And that's very important because it hones you down on the little universe of things that could be right.
No, I think you posed the question properly. I think we need to take risks, and I'm just sad that it's hard to do it nowadays.
Well, I think I don't want to be overly defensive about the NIH, but
I didn't mention it.
It's always in the background, and I couldn't agree more that there's a tendency in opposing research to be done to be cautious and be incremental and be within a safe zone. But one thing to remember about the NIH is they don't issue contracts; they issue grants to do research. And you're likely to have 4 or 5 years of support, and without someone hanging over your shoulder and saying, no, you can't do that experiment because you propose to do something else. And preserving that freedom while we work on the larger environment that makes people unduly cautious and makes the peers who sit on review sections hesitant to provide money for ideas that seem zany, if not just simply novel.
Harold, I'm not sure that everybody working in the field knows what you just said.
No,
I think that's
I agree with
absolutely
you,
true.
but I do want to emphasize it here because I think it is important to remember we're not, NIH is not generally, they issue some contracts, but most of what they issue are checks.
Right. Mm-hmm. Yeah, I was going to say exactly the same thing, that young investigators are, in fact, have considerable freedom to do what they want to do. They have to do it within the grounds of their budget and certain other parameters.
And I do think the NIH could do more. I've written about this and advocated that NIH Institute directors should be urging their study sections to support deviations from what people have done in the past, to get investigators who are applying for their first grants to do things that are not simply extensions of their highly successful work as trainees, to get people to understand that past accomplishments are probably as great a measure of how someone's going to do in the future as an idea that's well honed in the way it's described. So we need to be more adventurous, but there's also a large community of people seeking limited amounts of money.
Well, that's the problem in the end, right? There's a lot of competition.
Part of it,
yeah. So Harold, let's get together in New York and talk about science funding sometime.
Good.
It would be a good podcast. Yeah. Before we close, I just want to point out in this program, there's some really nice questions. We're not going to answer them, so don't worry. But I just think our audience should hear. They ask what detergent was used, and we kind of know already from what John said who named it.
I think we leaked that one.
What was the first virus used for large-scale commercial preparation, and why? I don't know the answer to that.
A lot of people would know that answer. I think.
If you were a Navy
people our
biologist,
age, people our age would know that answer.
What was? And then we have this. I like this one. Rank the following enzymes by their importance to science, as judged by the number of papers that mention them: RNA polymerase, DNA polymerase, protein kinase, reverse transcriptase. That's a tough one.
That's tough. Yeah.
I'd almost think protein kinase would be up there because of this.
So do I.
There are a lot of them.
Yeah, there are a lot of them too. All right. That is a special episode of TWiV recorded at 50 years of reverse transcriptase. And you know what, folks, for 100 years, I'm not going to be here. I'm sorry. You're going to have to get somebody else, and maybe I could build something that endures.
You need reverse aging.
That's reverse aging. That's right.
Exactly.
You can find all of the podcasts at microbe.tv/TWiV or anywhere on the internet. If you have questions or comments, you can send them to twiv@microbe.tv. And Microbe TV is a nonprofit 501c3. So if you like what we do, consider supporting us. And your contributions would be federal U.S. tax deductible. You can go to microbe.tv/contribute. My guest tonight from Caltech, David Baltimore. Thank you for joining us.
Very glad to be with you.
From Tufts University, John Coffin. Thanks, John.
Thank you. My pleasure.
Looking forward to number 4.
4.
Okay. And Harold Varmus is at Weill Cornell Medicine. Thank you, Harold.
My pleasure. Thank you, Vince.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the organizers of this meeting for having TWiV here. That's John Coffin, Steve Goff, Ann Skalka, Steve Hughes, and Hung Fan. And thanks to the guys in the booth for recording this episode. I get to dream. I get to come, sit down, and chat and leave, and it's all recorded. I don't have to bring any equipment myself. I just love it. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Thank you.